Here we go. All right, so this is really a brand new project that we started in the lab in collaboration with Adam Wende. Here goes. This is something that we've been doing. We've actually had a lot of fun doing this. It's been a lot of hard work that has gone into this, but it's turned out to be very rewarding. And upfront, I'm going to thank everybody in my lab and in Rajeev's lab who've collaborated and really lent a helping hand whenever needed to get this work done. This was spearheaded by two team members, Heva and Brandon, who are on the call, thankfully, to address any questions. And with, as I said, a helping hand from everybody in the lab. This would not have been possible at all without collaborations from Adam Wende and his group. In particular, Jenna, who's worked with Heba and Brandon for us to get this model up and going and validate it. And of course, a big shout out to Jenna. Congratulations for getting her postdoctoral award from the AHA. We've also collaborated with Dr. Sharafildin and of course, Rajeev's lab in moving this project forward. And I look forward to any insight that my faculty colleagues and anybody else on this call has to move this project in a nice direction. So what got us actually started were some of these staggering statistics. And in interacting with the Department of Pathology, my colleagues, and especially early on in just conversations with John Chatham, I kind of got drawn into the field of diabetes. And that's when I started looking it up, and it was staggering to note that, for estimates from 2 years ago, 10% of the population has diabetes. And in Alabama, these numbers are even greater. 15% of adults have diabetes, and 80% of these are overweight or obese, and 16% of those are morbidly obese. And in addition, about 37% of the adults in Alabama have prediabetes, which is typically an undiagnosed condition, which is then diagnosed later as diabetes. And most notably, metformin continues to be used as the first line of an anti-hyperglycemic treatment for those patients who are diagnosed with type 2 diabetes. And metformin typically lowers blood glucose, it inhibits hepatic gluconeogenesis, but it also has other effects, and many of those are still being discovered. Prominently, it inhibits the mitochondrial complex 1, and it limits the production of ATP. But importantly, as I said, it continues to be used as a first line of an anti-hyperglycemic treatment for these patients. And for our state of Alabama, which is dominated by rurality, it's important to note that metformin is covered by Medicare and Medicaid in all states of the country. So coming back to diabetes, the lifetime risk for a woman to develop diabetes who was born in 2000 is 1 in 2.5. For breast cancer, 1 in 8 women will develop breast cancer in their lifetime. And some women have to manage both conditions, breast cancer and diabetes. And this is where the problem becomes more compound and needs more investigation. It's also relevant to know that a significant number of breast cancer patients present with excess weight at the time of diagnosis. And these breast cancer patients who are diabetic and obese show an increase in mortality compared to those women who do not present with obesity or diabetes. So what we did was actually to use, our own cohort here of breast cancer patients at UAB. We used the I2B2 database, and we extracted data on female breast cancer patients who were diagnosed with breast cancer. So this cohort yielded us a little over 11,000 patients with the diagnosis of malignant breast cancer, and all of these are patients at UAB. And from these 11,000, we had close to 2,000 patients with the diagnosis of type 2 diabetes. So that's about 17.5%, so which is very close to what I told you about the state of Alabama being 15.4%. So here we had 17.5% who were diagnosed with type 2 diabetes. And of those, close to 80% were overweight or obese or morbidly obese. So this data was actually obtained in collaboration with Dr. Sharafuddin, who is an assistant professor in the Department of Medicine and is in the Institute for Cancer Survivorship. So she analyzed this data that have bubbled out from the I2P2 database. And right off the bat, you can see that overall, the survival of breast cancer patients who are diabetic here on the left-hand side is decreased compared to those patients who do not have a diagnosis of breast cancer, of diabetes. Moving here to the right-hand side, if you'll follow my arrow here, you'll see again that the 10-year survival probability for patients with diabetes is significantly lower than those without. Likewise, the survival for patients who are obese is significantly decreased. And if you put the two complications together, the two comorbidities together, you see that diabetes and obesity, a co-presentation of those, significantly decreases the 10-year survival probability. So all of these are breast cancer patients who were treated at UAB. Great. This is something that you would expect, but how do you study this? And this is where the challenge comes, because there is no reliable model that allows us to recapitulate breast cancer in a diabetic and obese host. So here is where we started collaborating with Adam and his lab and developed a mouse model of diet-induced obesity and hyperglycemia. We fed our mice. We started by feeding our mice with two different kinds of diets, different cohorts of mice. They were on a controlled diet with 10% fat, or on a high-fat diet, which includes a 60% fat. About a couple of weeks down, we injected them with streptozocin, and then a second dose of streptozocin a few days later. We continued the treatment and then injected the E0771 mammary carcinoma cells in the mammary fat pad, and then euthanized them about 21 days later. As you'll see here, the mice who were fed a high-fat diet here in the pink line showed definitely, these are fatter than the mice who were fed a controlled diet. And of course, these differences were significant, which told us that the diet is having an effect. As I mentioned, one of the drugs that is given as a first-line anti-hyperglycemic treatment is metformin. So we had a group in which we treated the mice with metformin here. And this was then this metformin was then administered through the end of the treatment. Shown here is some data on blood glucose levels. We see that the mice fed with the high-fat diet and injected with streptozocin had higher levels of blood glucose relative to mice who were injected with metformin here, the purple bar. You also see that the mice who were fed with metformin showed a slight decline in body weight. Not significant, but there was a trend towards a decreased body weight. When Jenna and Heba did the glucose tolerance test, you do see that metformin-treated mice have an overall improved glucose tolerance and a decreased area under the curve for blood glucose levels. What happens to the tumor then? So remember, I said we injected these mice with tumor cells, mammary carcinoma cells. These were labeled with luciferase, and which allowed us to image these mice by bioluminescent imaging. And as shown here on the left-hand side is the representative picture of these mice. And when you quantify the amount of the luciferase, which actually tells you the signal from the tumor cells, you see that the metformin-treated mice have smaller tumors compared to the mice who were just fed the obese diabetic diet and streptozocin. If you plot the overall luciferase signal, which indicates tumor growth as a function of blood glucose, you actually see that there's a very nice correlation between blood glucose and the tumor size, which tells you that the tumor growth is indeed impacted by blood glucose. So of course, if this is the Department of Pathology, so what do? We take these tumors, get them sectioned, and try to understand what's going on. And that's what we did. And shown here are just representative photomicrographs on the top and the data on the bottom. We first looked at the tissue for its level of O GlcNAc-ylation, because excess of glucose we would expect that it would get shunted through the hexosamine biosynthetic pathway and would result in elevated tissue O GlcNAc-ylation. And indeed, we see that the tissues are O GlcNAc-ylated in diabetic mice and are fat mice. Likewise, the tissues you can see have fat deposits, fat globules. These were very clear the moment we looked at them under the microscope. The tissues from these diabetic obese mice also showed a higher, an elevated signal for PCNA, proliferating cell nuclear antigen, and were much vascular. And metformin seemed to alleviate all of these attributes. As Adam mentioned, one of the topics that my lab is interested in is tumor immunology. And we've been working in the area of tumor immunology for the past about 7 or 8 years. And one of the things that we started doing is looking at macrophages. And quite simplistically, macrophages, as most of you may know, would exist in two forms. There are inflammatory M1 macrophages here on the right-hand side in blue, or there are immunosuppressive macrophages, which are alternately polarized, called as the M2 macrophages. We stained our tissue for these M1 and M2 macrophages. The M1 macrophages are marked by CD80, CD68 being a pan-macrophage marker. And we also stained these tissues for CD206, which marks the M2 macrophages. You'll see here that the tissues from obese and diabetic mice have significantly fewer M1 macrophages and seem to be enriched in these M2 macrophages. And metformin seems to alleviate this condition. And in fact, the tissue from these metformin-treated mice has many more M1 macrophages and significantly fewer M2 macrophages. So obesity and this hyperglycemic state are definitely impacting the population of macrophages that infiltrate into the mammary tumor. What we also did was to examine the architecture of collagen in these tumors. Now, why collagen? And it's important to look at this because collagen architecture determines or is associated with facilitating metastasis. While linearized collagen facilitates metastasis, a network collagen impedes metastasis. And that's what we see here when we stain this, when we stain these tissues for picrocereus red. On the bottom here is seen in bright light, and on the top is polarized light, are the same tissues in polarized light. And it's very clear here that the tissues from the obese diabetic mice show this nice linear collagen, which is actually quite distinct from what you see in the lean mice, as well as in the obese and the metformin-treated mice, which show these network collagen. And actually, it was very interesting that aligned with this, we saw that the mice from the tumors from obese and diabetic mice had these foci of metastases in the lungs, which were notably absent from the lean mice tumors as well as the metformin-treated mice. So this was interesting because this is actually not a metastatic line, but the conditions of obesity and diabetes seem to facilitate and seem to create conditions that can allow for metastatic seeding, or as we like to say, early metastatic seeding. So where do we go from here? Knowing that normalizing glucose, as we'd like to think that metformin does, is reconfiguring the mammary tumor microenvironment and decreasing metastatic seeding. What we would like to do is to analyze this tumor for comprehensively for a population of regulatory T cells, the type 1 helper T cells, the type 2 helper T cells, the activated CD8 positive T cells, as well as exhausted T cells, to know if the profile of these infiltrating immune cells might be impacted in an obese and diabetic host, and how this would be further altered by metformin. So this is our early and evolving research, and I'm happy to take any questions. questions, suggestions, most appreciated, because this, as I said, is brand
We
new
have two
research.
in the chat. The first one's by Pons. Do you know the status of the insulin receptor?
Do I know the status of the insulin receptor in these mice? So if you mean in the tumors, we have not looked at the tumors, and that's a very good point. We'll take a look at the insulin receptor in the tumors of these mice.
Okay. And then the other one's by Me3. Is there a concern about using luciferase since it is dependent on ATP? Because presumably the tumors are larger as well. Also is just looking at M1 and M2 and oversimplification of the macrophage diversity as far as subdividing those further.
Absolutely. So the good thing is monitoring the mice longitudinally by luciferase may be taken negatively, but the good thing is that at the end of it, when we look at the tissues and look for PCNA, it clearly indicates that those tissues are proliferating, that the cells within these tumor tissues are proliferating much faster. So actually, the good thing is that luciferase agrees and aligns very nicely with the PCNA. And we also monitor these tumors 3 times a week using calipers. So we have multiple measurements of proliferation and growth that we capture. And the second question is looking at the M1 and M2 and oversimplification of macrophage diversity. Definitely, yes. As one would imagine, these are just the polar extremes. Macrophages exist in a spectrum. But if you go to look at it in tumor tissue, there's no real way to capture this spectrum. And so it's quite accepted in the field that you would stain for these polar ends or the polar extremes of the macrophages.
And the final question by Joanne is TGF beta an immunosuppressive in the TME? Do you have any evidence of TGF beta signaling, TSP1 as an activator, TGF beta activator, et cetera?
No, we haven't looked at that, but we have the tissues. We also have serum from these mice, so we can definitely take a look, and definitely haven't done any TSP1 either.
Right.
And Suri says maybe worth looking at MMPs and collagen modifying enzymes. Definitely. Yep. The P4HA2. Yep. I
Well,
agree.
thank you so much for that. And we're a little bit over time. Fortunately, since I jumped in at the end, I can just cut myself out at the end if we run out of time. But I do want to continue on and introduce our next speaker, Dr. Rajeev-Samant. He also started his training by completing his master's degree in Bombay before continuing on for his PhD in molecular biology. Then came to the States as a postdoctoral scholar at Penn State before coming to UAB, and then in a similar trajectory, spending a little bit of time down in Mobile before joining us back up here, where he's also risen through the ranks as a tenured professor and also research scientist for the VA. Dr. Samant is really a great person to work with on committees. He's been very active in admissions and recruiting efforts, as well as having a very robust scientific research program, including also having 6 active grants for his PI or co-PI with nearly 100 manuscripts, and 2 very recent ones, a primary article in Cell Death and Disease, and an important methods paper in uncovering metastatic progression. And it's really been great to see this duo as they've progressed and both complemented each other, but also have very distinct scientific interests as well. And today we're going to hear Dr. Samant's differences in his research really focused on understanding the ribosome biogenesis in breast cancer. Dr. Samant.
Oh, thank you for that fantastic introduction, and hello everyone. Well, I have never given a scientific presentation following Lalita's presentation, so this will be interesting. So hopefully I will keep the tempo and but change some gears here. So hopefully you can see my screen. I'll be speaking with you about my lab's recent interest. Our group has been focusing on this for maybe past 6 years. The reasons is a story, so I won't go digress and tell you a history about it. However, I will tell you about our lab's interest in understanding the ribosome biogenesis and nucleolar biology in cancer. And my lab's focus has always been solid tumors and breast cancer and progression of breast cancer. So I don't have any conflicts. And first of all, I would like to share with you that Shannon graduated in February. Most of what I will present is a part of her thesis, with a lot of help from Brandon and Sarah and Heba. You heard about Heba and Brandon's work in the previous presentation also. And Amra is a new graduate student. I hope he will contribute to wonderful stories that I'll get to share with you. And of course, the collaborators from Pathology are always on the forefront. I collaborated a lot with Dr. Shi Wei and of course Lalita's lab. And through the College of Medicine with David Schneider, who is an expert on POL1, and I will talk to you about POL1, RNA polymerase 1, and Jim Mobley for proteomics, and DQ, Dongkwon Chen for informatics, and Akin also for informatics. So overall research focus is the nucleolus, and I welcome questions in all directions. This is a new field for us. Nucleolus, as many of you have known from textbooks, is a small little body within the nucleus. And all that I studied, at least in college, is it makes ribosomes. It's a sensor of tumor microenvironment. So it senses hypoxia. We published that work also. I'm not talking about that today, maybe some other time. And what I'll be sharing with you is its role in responding to wind signals, or its components responding to wind stimuli in cancer, at least. So here's the nucleolus and the green dot, very focused green dots, no puns intended, but we use a lot of microscopy. You see a little bit of green color here, cytoplasms, and these bluish, greenish things are nuclei. And nucleolus is formed at the specific locus on acrocentric chromosomes, chromosome 13, 14, 15, 21, and 22. And these are repeats, like telomere repeats. Ribosomal DNA is in repeats, and there are hundreds of such repeats. And if you use electron microscopy, you can see that nucleolus has a structure, and this is a very hot topic in research, believe it or not, that people care about how nucleolus is formed, and it's actually a globular conglomeration of proteins. There is no membrane binding to this, so this is not an organelle. And it has a fibrillar center, dense fibrillar center, and granular center or granular region. So this is a very, very zoomed in image of nucleolus. And a lot of things, as you can imagine, happen in the nucleolus. So what is that to do with cancer? Well, after microscopes were discovered and people could see cells and people could see human cells, one of the prominent features that pathologist in 1800, an Italian pathologist named Giuseppe Panini, discovered that these dots are characteristic of nucleolar of cancerous cells. And it was not long for people to realize that these dots in cancerous cells are more numerous and they're irregular. So you can see this zoomed in version of an Agnor staining of breast cancer nucleoli here in black. There are more black dots and more irregular black dots in aggressive tumors versus fewer black dots and more circular black dots in more normal or benign samples. And this correlates with poor prognosis. So increased Agnor staining indicates bad prognosis. So really, it's a sort of a prognostic indicator, so to say, for solid tumors, including breast cancers. However, not much was known beyond that. People didn't study the relations of solid tumor biology to nucleolar biology, and I thought that's interesting. And the observation that drove this home for us is this triple negative breast cancers, and many of you know that there are molecular subtypes of breast cancer, and I will just stick to hormone receptor negative, triple negative, and hormone receptor positive, and I will call them as non-TNBCs, where we have clubbed lots of samples. But grossly, you will see lots of nucleoli in TNBC specimens versus non-TNBCs show less nucleoli, and they show more normal-like phenotype. And if you count meticulously, which Shannon and Sarah did, you will see that there is a statistical difference in nucleoli per nucleus in TNBCs versus non-TNBCs. So that's really interesting, and we wanted to see if this holds true in the various cell lines that we use in the lab. And here are 6 TNBC cell lines and 6 non-TNBC cell lines. And by non-TNBC, I club luminal and HER2 positive. But we see a difference that kind of recapitulates what we saw in TNBC patients. So as I mentioned, nucleolus is a conglomeration of proteins. There are about 1,365, that was the last count, proteins confirmed to exist in nucleolus. They are not static. Nucleolus is very dynamic. As I said, there is no membrane. So proteins come and go. There are some proteins that stay there. So it's really, really interesting mishmash of proteins, and many of them are doing ribosome biogenesis. They make ribosomes, but that's up to 400 proteins there. So there are others that do stuff that we have no clue about. And we thought that was interesting. So Shannon took a very remarkable task of profiling nucleolar proteomes of breast cancer cell lines. So we generated until by far the biggest nucleolar proteome collection for breast cancer. So we had these 6 non-TNBC cells and 6 TNBC cells, nucleolar proteome analyzed in 2 independent passages, so biological replicates. And here is just a summary of all that massive data presented as similarity matrix. We got this in collaboration with Akeen across the hall. He actually previously worked with Broad Institute and taught us how to do that. But essentially what it shows is the same cell line nucleoli are identical. So that's the brightest red color here. Whereas most of the non-TNBCs cluster together. So most brighter red square here versus most TNBCs, this square, cluster together. And some cells have some overlap with TNBC-like nucleolar proteome, whereas some TNBC cells have some non-TNBC-like nucleolar proteome. So that's expected, but what was gratifying to see is TNBCs, for the most part, have a distinct nucleolar proteome. So that's really remarkable. So what is there in that proteome? And we had this on an average 12 to 14, 1500 proteins with significant score in the mass spectrometry. So we found out that there are 129 proteins roughly common in non-TNBC cohort that's not in TNBC cohort. And in TNBC, there are 145 proteins. Since we cared a lot about TNBC because that's a more aggressive cancer, we were biased to focus on TNBC. And we quickly realized that most of them are transcriptional targets of a transcriptional cofactor, beta-catenin. This is an interest in my lab for a long time. We study Wnt beta-catenin signaling pathway. So it was kind of natural for us to get drawn to see so many targets of beta-catenin in the nucleolus. And there are 108 of them specific to TNBCs, and 50 of them are actually confirmed nucleolar proteins. So if you guys know, there is a human protein atlas, so you can go there and find out where protein of your interest is located. And some of them are nucleolar, so these are confirmed nucleolar proteins. So that's a big portion of proteins that are regulated by Wnt signaling cofactor. So we wanted to know more about this. And it's important to emphasize that it is known for a long time that Wnt signaling is important in TNBC biology. People have been struggling to target breast cancer and TNBC with Wnt inhibitors. We won't talk about that today much, at least not as a part of what I will discuss with you. However, here to emphasize that finding about beta-catenin, you can see beta-catenin has two cellular locations, mainly, or three rather. It could be membranous, it could be cytoplasmic, or it could be nuclear. And when it is nuclear, for simplicity's sake, it's bad. And TNBCs show a lot of nuclear beta-catenin. Those people who work on colon cancer know that this is a typical indicator of poor prognosis and aggressive colon cancer. In breast cancer, this staining is not that stark. So you have to actually uh, do meticulous counting of nuclear to membrane ratio, and you see that there is high nuclear presence in TNBCs, low membrane presence, versus non-TNBC mostly show membrane presence, low nuclear presence. So what does this have to do with nucleolus? So if you take the agnor staining and combine all beta-catenin staining with membrane and nuclear stain and combine with agnor stain, you will see that these non-TNBC cells, which have lower nucleolar number, cluster here with low nuclear to membrane score of beta-catenin, whereas the TNBC cells cluster here show higher membrane nuclear to membrane ratio and more agnor score. Um, the number doesn't seem to be a lot. It goes from, say, 1 point something to 2 point something. But believe me, when you have those many ribosomes generated in the cell, it's remarkable, and that's unusual for cell biology. So what happens if we try to tinker with the Wnt signaling? So we used a reagent ICRT14 and at a concentration that's much lower than what can kill the cell. So if you use a lot of this inhibitor, it will kill TNBC cells. But we used it at 5 micromolar, which we standardized, does not kill the cells, keeps over 95% cells alive or somewhere there. And you see the nucleolar staining comes back to what looks more normal like. Fewer numbers compared to more numbers in the same TNBC cells upon inhibition of Wnt signaling. So we said, what happens to the nucleolar proteome? And Shannon again captured ICRT treated couple of TNBC cell lines, compared it with non-ICRT treated. And one of the proteins that stood up was LAS1L. And why did it stand up for us? Is because this was the only common protein that was enriched in TNBC nucleoli that was suppressed by Wnt signaling in after treatment of those TNBC nucleoli. So that was remarkable, and we focused on that and looked at TNBC data as to what, if at all, has a relevance for LAS1 in breast cancer. And high LAS1L staining, at least transcript levels, are seen in breast cancer patients, and that is correlated with their survival probability. So encouraged by that, we wanted to know more about LAS1, and LAS1 is actually related to a rare disease, a mental retardation syndrome, MRXS6 or WTS, is attributed to LAS1 mutations. How mechanistically is not known, but it's seen as a genetic marker of those diseases. LAS1 is a member of a complex that processes the 47S pre-ribosomal transcript. So the ribosomal RNA, 18S, 5.S, and 28S, are actually transcribed of a one message, and then they are clipped from that, spliced, and then the ribosomal RNA is formed. These three, 5.S, 28S, and of course 5S comes from a different mechanism, and we won't talk about that today, but contribute to this large ribosomal subunit 60S. The small is formed by 18S ribosomal RNA. And the 60S formation is regulated by LAS1L. That was known from yeast studies as well as some human studies. We checked if LAS1L knockdown changes the profile of ribosomal abundance, ribosomal subunit abundance using ribosome profiling, using um, gradient centrifugation, sucrose gradient centrifugation. And you can see that 60S ribosomal subunit is present in the control cells, the control TNBC cells. And upon knockdown of LAS1L, the presence of 60S is reduced, whereas 40S is pretty much the same. And if you calculate this a little further, the area under 60S curve upon area under 40S curve shows a significant difference. That means 60S production is reduced, not stopped, it's reduced. The cells are still alive and functional. And is LAS1L in breast cancer really localized to nucleolus, as Human Protein Atlas says? And this wonderful staining essentially confirms fibrillary is a protein that is exclusively nucleolar, so is a marker of nucleoli. LAS1L is pretty much concurrent overlapping with fibrillary, and here is the overlay and zoomed in picture, and you see the same thing in TNBC patients. So LAS1 had never been profiled for presence or absence in breast cancer at immunohistochemistry level. So we did that. It's seen predominantly in the nucleoli, and in TNBC there is much higher staining for LAS1L. And what happens to these cells if LAS1L levels are not there or compromised? So you can see our silenced cells show a little bit of LAS1. Maybe that was a blessing in disguise. So the nucleolar number gets reduced compared to the control, which is great. And the cells don't invade or migrate. So these are invasion and migration studies using Boyden chambers. And more so in animal models, in xenograft studies, the tumors grow slowly. If you don't have enough ribosome biogenesis, you would expect that. And they, most importantly for us, the percent metastasis-free mice was more for LAS1L silent cells. So there were less metastasis in those mice. So the tumor showed less staining. And diagnosed staining shows what we predict it would, more regular nucleoli and fewer nucleoli. So normalized nucleolar biology. So that pretty much concludes my talk, and that is what I wanted to share with you, is the exciting finding about nucleolar biology and ribosome biogenesis relevance to the breast cancer. This is, to our knowledge, the first report that says that. So thank you. And any questions?
Well, thank you very much. Very different aspect of cancer and very exciting new findings. You have two questions in the chat, one from Vishnu. Nucleoli Agnar score associated with aggressiveness, whereas in lymphomas, Burkitt type, other aggressive lymphoma, where do you see the nucleoli? Like, is it common to see those?
So I have not studied nucleoli in lymphomas. I have heard from Vic Lin, who actually is now I believe he's a faculty, but he was a fellow working in Lalita's lab, and he always said about leukemia and lymphoma showing prominent nucleoli. So I need to look that up more, and I'm sure there is a lot more biology to study there. So
Rajeev, this is Vishnu. In the Burkitt lymphomas, what I meant to say, we don't see any, we do not see nucleoli. In fact, the chromatin is smooth. And it's a very unusual, but also when you look at the proliferative rate, it is close to 100%. If you do the Ki-67, it is almost 98 to 99% in those lymphomas. They're very, but we don't see nucleoli. We see fine chromatin, fine to distributed chromatin. So that was my observation. Obviously, we did not do Agnar scores, so I have no way to correlate that. But that's what I was saying. Sometimes aggressiveness may not have any nucleoli. That's what I was saying.
Interesting. That's great information, and actually, I will like to look more and learn more about that from you and others.
Thank you.
And then just very briefly, Surrey highlights two new findings that relate to some very interesting and very recent discoveries with EZH2, the histone methyltransferase, specifically a new role in binding to rRNA and actually methylating the RNA. So a different type of epigenetic regulation, as well as some of the combined analysis with UALCAN in these gene expression and one of the GS or GlaxoSmithKline inhibitors. But with that, I will wrap up because we can all continue this conversation since we're all on campus. So thank you very much for that. And I will turn over to the last speaker, Dr. Alan Bryan. So Dr. Bryan received his Ph.D. and medical degrees, both from Harvard, before continuing on with his postdoctoral fellowship and residency at Beth Israel. We were then able to recruit him to UAB in 2015, where he really exemplifies the physician scientist, really combining this interface of laboratory medicine practice and clinical informatics to really bring together large analyses for specialization of microbiology, chemistry, and molecular pathology. He's also very active in teaching, focused on sharing his knowledge in microbiology as well as informatics. And he translates those interests into publications, including publications in Laboratory Medicine, Journal of Clinical Microbiology, and PNAS. He's held a number of interesting hats. The one that stood out most to me was his director of the Infectolitics project, which was sponsored by the CDC, and also his award of the Presidential Fellowship Award a few years ago. But to not take up any more of his time, I'll pass over the mic to Dr. Bryan as he tells us about the new COVID-19 testing system. Thank you, Dr. Bryan.
Thank you. And we're here to talk specifically. This is a development that was made just in the last few months across the street at the VA, where I am the microbiology, they call it consultant, but it means section head.
So Alan, it's showing, yep, there you go. Perfect. Thank you.
So this came out of a national initiative that the VA headquarters gave out last October, where they were desiring to expand the robustness of our PCR testing capabilities nationwide in response to the anticipated third wave of the pandemic. The mandates included insourcing as much as possible, as well as developing in-house capabilities, both not only for the robustness for the immediate third wave, but also flexibility for variants and other waves coming down the pike. And this is part of a more general surge where we were pushed out in quite a quick manner. We pushed out the Biogenesis Now point of care card for lateral flow assay. We pushed out a new version of the Cepheid PCR assay, which we had already been using. We installed and pushed out a BioFire, which has a panel for respiratory disease differential diagnosis using an array of PCR tests. So that allowed us to quickly distinguish people who were symptomatic from COVID and being symptomatic from other respiratory diseases. And we instituted an antibody testing platform, which is a more straightforward system. However, there was serious concern about the robustness of the Cepheid platform. And this is due to a bunch of acute problems in the supply chain, some of them well upstream of Cepheid itself, which led to there being an uneven and unpredictable availability. They were rationing these pods for all providers, and that meant locally we were triaging and rationing those testing kits to only the things that needed the most immediate testing. And this highlighted just how inflexible the Cepheid platform was. Now, fortunately, Dr. Sixto Leal had implemented our GuideSafe in the fungal reference lab, and that provided us with a terrific safety net. And thank you, Dr. Leal, for developing that. I was out having my daughter being born, so it was a fantastic job. I came back and everything was already rolling. However, the VA saw that as only a starting point, and they wanted to use that as a safety net while we bridged over to our new capacity. That new capacity was enabled. I'm going to briefly go over some similarities in molecular testing. Pretty much all nucleic acid methods have the same 3 similar steps of getting a hold of the nucleic acid, amplifying whatever target you're looking for, and then detecting and analyzing the signals that come out of that reaction. What most labs implement are what we call closed platforms. That is to say, once you have entered the sample, they are otherwise self-contained. And that includes the Cepheid and the BioFire systems. They have different technologies on the inside, but the key point is that they are all in one solutions. Once you have sealed it in, all the extraction, the cycling, and the detection all happens within the same set of chambers. That makes it very easy to set up and very easy for quick implementation, and it's quick to train people on, but it's very hard to modify. You are basically stuck with whatever panels and primers and testing modalities that the manufacturer provides. However, because of the similarities between all of these things, where there are standard methods to extract, similar mechanisms for the amplification, and common markers used across many different mechanisms for doing detection and analysis, that means that the industry makes a lot of instruments and automation platforms that can be somewhat mixed and matched. They're flexible, can be run with many different reactions, they are compatible with each other, and they're even partially interchangeable. So we wanted to leverage that the same way that UAB did by producing an open platform. And an open platform uses different instruments and different techniques within the instruments. So for instance, Dr. Leal has a silica membrane elution system, and then uses primers from Thermo Fisher to make the analysis. The key point here being that there's more than one instrument and there's more than one component. Now, the price you pay for that is that you have to do a lot more of the setup yourself, and you need a dedicated space because now you don't have a sealed system and you have to avoid contamination as you transfer things from one instrument to the other. The advantage, though, is that it's easy to modify whenever you need. So what did we do at the VA? We have developed a novel protocol, but based, thanks to this modular system, on already known and already proven methods. So for our extraction step, we are utilizing a proteinase K elution-free protocol that was pioneered by the teams at Yale University in their outpatient and commercial testing platform, Saliva Direct. This has the advantage of being able to be used on other types of samples. We are not using the saliva method. We are using regular nasal swabbing, but the use of saliva highlights just how robust this extraction process is, because prior to this protocol, there were a lot of attempts to do testing in the early months from saliva. It's a much more convenient, certainly much more pleasant experience than getting that nasal swab. But there was no way for the elution techniques to sufficiently isolate the virus, DNA and RNA, from the sample. Fortunately, this system allows that to happen, and it even does so in a simpler method. There's no elution step because the proteinase is going to dissolve all the stuff that you would have eluted. And since it's been tested out and used by Yale, including they were doing part of the testing for the NBA bubble. So we know quite well that this is going to be effective. For our cycling, we're using the same general types of hardware as being used in the fungal reference lab, but we are using a different set of primers. These were originally developed over in South Korea by a group called OSANG, and they are in fact the worldwide gold standard. So all the other COVID molecular and antigen-based testing protocols that you have seen used were all tested against this. This is the reference method. So we are quite confident in its capabilities. And so we are putting these two together with a slight automation here. A good bit of this is a manual step right now, while using the proteinase K treatment to boost our detection capabilities, and then feeding it into an ABI 7500 thermocycler using the OSANG primer system. And to our knowledge, while these are all proven components, this is a novel combination. We are sure this is a novel combination within the national VA system. We believe it is a novel combination throughout the United States. I just want to highlight what has been done here, because when we started this project in November, we had no lab space set aside for any sort of molecular testing in molecular micro. We had to start with a bare room. We had no techs that had any training or experience with the equipment we were using. And I was the only one who had had any exposure to open platform molecular testing at all. No one else had any previous experience. The lab was short-staffed, and we were trying to do all of our regular services during the third wave of the pandemic. And despite all these challenges, we got equipment in December 15th. We got things set up over the holiday period. By January 5th, installation of the instruments was complete. 2 weeks later, we got our procedure assembled, and we were doing our first trial runs. Less than a month later, we completed validation, and a couple of weeks after that, we went live. So for that whole cycle, it was 4 months from planning to go live, 3 months from first hardware to go live. And given the number of challenges we have, I am just so proud of the team at the VA that has made this possible. I do want to acknowledge the leadership that gave us lots of support from the director on down. The deputy chief of staff and the chief of path were really deeply involved in the day-to-day nuances of working through the supply chain problems, the installation problems, and everything. We got a lot of help from our partners at VetExcel, but the biggest thing that I have to shout out to is the teams, especially the supervisor, Teresa Horton, and our lead tech, Ikea Moore, who put in tremendous overtime making all of this work and learning techniques from pipetting to isolation and extraction on the fly. And it has been absolutely a pleasure and a privilege to work with. So just to overview here, we have moved some of our operations. We still use the BioFire for our diagnostic platform, our differential rapid testing. We still have Cepheid available for the very few cases when we need an answer in 1 to 2 hours. Like Dr. Leal's operation, we are testing a certain number of batches per day. At the moment, we have 1. As automation proceeds, we will be increasing that ultimately to 3 runs per day. But we are utilizing a novel system and that is open and is therefore flexible for testing. And with that, I will look at questions, and I see that there's one in the chat already. See, with various variants appearing, is there a way to develop specific tests to find out which one is present or if there are multiple variants present? Does not change anything as per quarantine, of course. That is possible. We are not doing development of primers. There is, Dr. Leal's lab does have a means to detect specific variants, and in fact has detected some of the variants that have been in the news in Alabama in low amounts. Once those become sufficiently mutated as to reduce the effectiveness of our current assay, we will be able to rapidly replace those primers with new sequences that are just altered just enough to make sure that our assay remains as sensitive and specific as when we initially implemented. Does that answer your question?
I think it did. I mean, I had the same question actually.
Thanks.
So
Maybe it may become important as per vaccination concern down the line.
Vaccination consult, you say?
No.
Sorry.
As per vaccination concern, like the present vaccine may not work for some variants. That is when probably tests also become important.
Correct. So as we approach herd immunity for the original strain, then the spread of variants will become more, first epidemiologically important, and then clinically important. And so the whole idea behind moving to an open platform was that we were less dependent on a single manufacturer or a single supply chain. We can use whichever primers are being produced in sufficient quantity and with sufficient quality to meet testing needs. So that is the big advantage that we have. The question here in the chat, what are the diagnostic performance compared to other platforms? So we are meeting or exceeding the performance from the Cepheid. We in fact have had a couple of discrepancies between the Cepheid and the OSAG system in both directions, and we are able to give both better sensitivity and better specificity. We've had a few instances where the Cepheid made a very low signal detection, but we were unable to modify its algorithms regarding the strength of signal required to make a detection. And we were able to show with multiple platforms, we ran it on our system twice and the BioFire, and they all came up negative. So it's not just that it is more sensitive, it is also higher reliability in many avenues.
Great, thank you so much. And I guess the cancellation worked out perfectly for us, because it seemed like 3 talks were the best way to divvy up the time today. So thank you to all 3 of our speakers. We heard the intersection of diabetes and cancer. We learned new mechanisms for cancer and ribosomes in cancer progression, and great application to really finish off the trio and how we can move forward in combating the pandemic. So thank you all for joining.
I think we had our right quickly from Sixto. They are not only do they have the particular variants, they are now working on a pooled method for doing novel sequencing de novo, and therefore being able to detect even new variants that others have not worked on. So that's fantastic, awesome. And I'm looking forward to hearing that talk.
Well, great. Well, thank you all, and please join us next week. I'll have a timer for the trainee speakers, and we'll hopefully get through 5 of them next week. So please join us, and thank you again to our 3 speakers today. Have a great day.
Thank you.
Thanks, Adam.